So I'll make this little screencast here about enzyme kinetics discussion. This is based off of our work Thursday and Friday of this week, the first go around here. I want to just make some comments about some things that I was observing and things that different groups are having a hard time with, and maybe this will help you guys get ready for next week a little bit. So the first thing here, this is what was on the board to the right again. What we were having you do in class with your groups was first, generate a list of those variables that could be affecting the initial rates of the reaction, and looking at the background and introduction stuff, you guys should come up with a list of that. Then having you sketch graphs for the predictions for each variable showing those effects. And so we had what was going to be over here and what was going to go here, and then what would the curve look like. Okay, so what are we trying to measure in the lab there? Over what conditions are we trying to measure it? And then what would your predicted curve look like? Okay, then designing a controlled experiment, meaning that you have a control group and then you have experimental groups to which you're going to be comparing, or with which you're going to be comparing the control group stuff there. And so trying to get you just setting it up here today to, again, show proof of concept, work out any kinks or problems, missteps encountered along the way, so that next class period, that is Monday or Tuesday of next week, as soon as you come in, you are off and rolling and ready to go. One thing that was maybe not apparent to everybody is that this is the reaction that we are all looking at. We're all looking at the same exact reaction, the same substrate here in peroxide, the same enzyme, and we're going to be yielding the same two products over here. And so we're going to be measuring the rate of this reaction happening by how quickly this oxygen gas is produced, right? So on the computers, you're going to be measuring the pressure changes over time. So you're going to be getting some sort of line like this, and again, then you need to take the slope of this best fit line and then plot that later on. So you're going to get lots of lines like that on the graphs. Okay. Some people are having a hard time just with the setup, so here's a picture again of the setup. In the syringe right here is going to go 3 milliliters of the substrate, right? That is the hydrogen peroxide. Now, making sure that everybody is using the 3% hydrogen peroxide solution, and this is what's in the brown bottles. And we had some confusion with that, different groups using different substrate concentrations. The only group that's using different substrate concentrations are groups that chose to vary the substrate concentration, in which case you're going to be, this is not true for you. You're going to be using different peroxide solutions there. So the substrate, the peroxide, goes in the syringe. This is the stopcock valve here. I'll talk about that in just a second. We had some issues with that. This tubing right here is hooked up to the pressure sensor. So this goes to the pressure sensor, which is hooked up to the computer, right? We had some groups screw up which they were putting the syringe on top of here. I don't know what happened there, but so just make sure you have that figured out. Then in the tube down here goes 1 milliliter of the enzyme, and we're going to be using the 400 international unit concentration of enzyme. Right? We had some groups using a 600. You weren't paying attention back there to which one you are grabbing. Right? So everybody's using 400 international unit there, and 3 mLs of the 3% peroxide up here. Unless, of course, you're varying enzyme concentration. If you're a group that's varying your enzyme concentration, then, of course, you're going to be using different enzyme concentrations down there. But here's something else that I tried to highlight with every group, is that also what goes into here should be 1 milliliter of distilled water. Right? 1 milliliter of distilled water goes in there as well. Unless you are a group that's going to be varying the salt or the salinity of that solution and or pH. Right? So instead of putting distilled water in there, you'd be putting your various salt concentration things in there, 1 milliliter of that, and or putting in your different pH buffers in there. In both of these cases, you would then sort of be mixing them, and I'm guessing here that 3 minutes might be enough time to sort of mix that up and have any effect down there. All right. So there's the setup. Let me just go talk about this whole stopcock valve thing here. So here's a close-up of the stopcock valve. We had one group that was having issues, with this part right here. The syringe screws onto this stopcock valve right here, so make sure that there's a tight seal there. This stopcock, here's the stopcock valve, and you notice that this is closed. This is in a closed position. In fact, you can see the little hole right here. If you can look through there and see the hole, then you know that that's closed. Right? So what you do then is once you get your substrate in there, you're going to open the stopcock valve here. Right? And so there it is in the open position. And you can see that you cannot see that hole there because that hole is in line with the substrate. So you'd be able then to squeeze the substrate down into the test tube down there. After you squeeze that or put that substrate in there, then you're going to go back and you're going to close this valve, right, as the reaction happens. Right? So you want to go back to that position. Right? So we had some groups not understanding what was going on there, right, and messing that up a little bit. So you want to close that valve so that any oxygen gas that's being produced is going to come up and go this way to the sensor. If this is open, then you'd have some gas coming back up here, and your whole plunger thing is going to just burst out the top of the syringe there. So that's not what you want. So you got to make sure that that is closed so that your oxygen gas again goes down the tubing there. Okay. If there are other questions about things, then just shoot me an email, and I'll try to help you out. For the temperature people, groups that are varying temperature, we had a big issue with this. I moved all the water baths to the left side of the room, and so I think what we're going to do is this is the hardest one to set up. All right, so for temperature groups, let's go with a 4 degree. Let's go with a 10 degree C. We'll go a 22 degrees, so that's a room. Right? So these are all colder to room. We'll try to get a 37. Let's try to get a 45, and then let's try to get sort of like a 55. So 1, 2, 3, 4, 5, 6 different temperatures there. We'll get these baths set up, right? And we'll go from there. We'll see if we can, if that's going to be good enough. So because we had lots of groups trying to set this up, and everybody at different temperatures, and getting these water baths to be, especially these warmer ones here, set up early enough and long enough, or early enough so that the water gets up to that temperature is sometimes a little bit tricky. So let's standardize the temperature there for all those groups. Okay, and I think we'll just go with that, and I think that's it. So if there's questions or confusions, let me know.